Hello folks, I'm going to do a quick video for you on presynaptic inhibition and facilitation as ways of either inhibiting an action potential being created in a postsynaptic cell or facilitating an action potential being created in a postsynaptic cell. So we're really kind of focused on these two learning outcomes here. Um, I'm going to show you a couple of wiring diagrams and review a couple concepts from signal transduction, and before I show you each one of these examples. So what I've drawn here is a few different neurons, and one thing to recognize about the wiring of the nervous system is that it is complex, and there are typically far more connections even than I've drawn here between neurons. So I've drawn a presynaptic neuron number one here, just a synaptic terminal, right, ending on a synaptic terminal of another presynaptic neuron. So this is presynaptic neuron number two. And just to remind us, we'll say that this is a pre, this is also a pre, and then this is our postsynaptic neuron. So presynaptic neuron 2 actually does synapse on the cell body of this third neuron and this postsynaptic neuron. Okay, so this is the wiring. Now, a couple of reminders about uh, signal transduction is, in general, there are many vesicles in the presynaptic terminal with neurotransmitter. The arrival of one action potential typically causes a small subset of those vesicles to fuse with the plasma membrane and release neurotransmitter. So in general, I'll just say 1 AP leads to fusion of a fraction of vesicles, right? So just a fraction of the vesicles that are there typically fuse. And then those open some of the gated channels on this postsynaptic cell. So a fraction of those total gated channels actually bind to the amount of neurotransmitter that's released and open. So a fraction of vesicles release neurotransmitter, and that means that a fraction of these ligand gated channels open. Okay, so that's one thing to just keep in mind. Another is that, and I may have mentioned this before and certainly discuss it in a future video, that what this postsynaptic cell does is really determined by the receptors that it expresses on the plasma membrane, right? And definitely the neurotransmitter seems to be important, although perhaps this is just a pattern that we've picked up on as we look across the nervous system. But so it's sort of the twin things of the neurotransmitter and the receptor that it binds to that's going to determine what happens to this postsynaptic cell. So in some cases, and all the cases we've looked at so far, acetylcholine release from a presynaptic cell causes a depolarizing stimulus in this postsynaptic cell, or an IPSP in this postsynaptic cell, an inhibitory postsynaptic potential. Sorry, I said that backwards. An excitatory postsynaptic potential, right? It moves it to threshold. But there are cases where acetylcholine binds to a different type of receptor, and when that receptor uh, becomes more permeable, and it's a gated channel. And when that channel becomes more permeable to its ions, we get an IPSP, or a hyperpolarization in this cell, moving further from the threshold rather than towards it. So it really depends on the two things, the type of receptor that's involved and the neurotransmitter. Okay. So in terms of how these can work together to either inhibit or facilitate transduction of an action potential in a downstream cell or a postsynaptic cell, is you might imagine we'll just use this single neurotransmitter ACH, right? So you might imagine an action potential arrives, for example, in cell number 1, right? And we'll think first about how this cell might inhibit a action potential in this postsynaptic cell. Right? So cell number 1 fires an action potential. When that action potential arrives, it releases its acetylcholine ions. So here's those ions diffusing across. They bind to, let's say they bind to a gated channel, and the gated channel they bind to is a calcium channel. But what they do is to uh, keep that calcium channel shut. So they actually block the calcium channel, for example. Right. So they block calcium channels. So they block the influx of calcium. Right. So we get calcium trying to enter, but not entering. Now, let's say that happens a split second, just a millisecond before an action potential arrives in this cell. Right. You know, typically that what happens with signal transduction is that calcium enters and facilitates fusion of those synaptic vesicles, right? And that's what happened in this cell. But this particular neurotransmitter receptor combo resulted in a blocked calcium channel, right? And so now when another action potential arrives here, we do get calcium trying to enter, but again, they're going to encounter some of these blocked channels, right? And so that means that when an action potential arrives in this cell, if it happens a millisecond after an action potential arrives at this cell, a smaller amount of calcium is going to be released, right? If a smaller amount of calcium is released, a smaller amount of neurotransmitter is released, right? So I'm drawing fewer neurotransmitters being released here than here, because of this blockage of the calcium channels on this presynaptic cell number 2. So that means a smaller number of these, say they're sodium channels, right? Smaller number of sodium channels will open, and we'll get less influx of sodium, and less than normal. So this is an inhibitory this is presynaptic inhibition, because the outcome is that the EPSP that's normally produced in this postsynaptic cell is smaller than normal. And that's because less calcium was able to enter this presynaptic neuron, less neurotransmitter released, and less bound to this, or less, bound to this sodium channel. So less positively charged sodium flooded into this cell. Right? Okay, so you could, let's summarize this here with presynaptic inhibition is where less neurotransmitter is released from the presynaptic cell due to some sort of inhibitory mechanism here where, for example, calcium channels are blocked. So due to some sort of inhibition or reduction of signal transduction. We'll just say it sort of very simply as signal transduction. This is just one example of it. Okay, so that's in presynaptic inhibition. Might make sense to actually look at this on some axes we're familiar with, where we're measuring the polarity over time. So we've got millivolts here on the y-axis, time on the x-axis. Here we are at resting potential, -70. Here's perhaps our threshold potential at -55. And under normal circumstances, you would get an action potential in neuron number 2, right? So we're actually looking here at neuron number 2. You would get an action potential sweeping down here, and bringing this cell, right? Let's say we're measuring it right here, for example, right before. So we're measuring it right before signal transduction, right? Bringing this cell to threshold and continuing the AP this short way down before it actually gets transduced, right? Bringing it to threshold. Now, in this case of presynaptic inhibition, imagine we get this action potential arriving, blocking some of these calcium channels in this synaptic terminal. What would this look like? Well, let's do a little red line here, right? We'd be operating here at -70, and then due to reduced calcium influx, that means there's a reduction in this, for this very short period here, there's actually a reduction in the amount of calcium that enters. So there's a reduction in the depolarization that takes place. So maybe it only gets here, right? So there could be a couple of things that happen. It could actually stop the, or sorry, it could really reduce the amount of neurotransmitter that's released. It could even potentially, if depending on where this sort of synapse happened, it could even stop the eventual transduction of this signal. In our case, it simply reduces, right, the depolarization that occurs. If we were to look at the same thing that happens here in this cell, it would basically look the same, right? So whether we're looking at cell 2 or cell 3, under normal circumstances, the entry of this sodium through voltage gated channels brings this cell to threshold, and we get a reinitiation of an action potential, right? But in this case, we might get less entry of sodium, and it might not bring it to threshold. And of course, the last thing to remember about this is, right, we're talking about charge entering and, of course, propagating, diffusing passively from this point of entry all the way to here, which is our trigger zone or our axon hillock, right? This is where, of course, all of this positive charge is sort of summed up to see, does this get to threshold? And if it does, then we get the firing of an action potential here. But do remember that this influx of sodium here just creates a graded potential that sort of spreads passively here. Okay, now let's look at facilitation. Okay. So with facilitation, sort of the reverse thing is happening, right? So I've just kind of erased what happened here. I added a couple more channels here, just to remind you that only a fraction of these have opened in presynaptic inhibition, but there are, of course, more that didn't, and that's because of a reduction in this acetylcholine that's released. But in facilitation, we have the opposite happening. So we still imagine an action potential arriving here at split second before an action potential arrives at nerve 2. And we get the influx of calcium that motivates the fusion of vesicles with the plasma membrane. And so we get our, we'll still say it's the same, right? Acetylcholine released, but this time the acetylcholine, and this is totally based on the receptor, right? Just the structure of the receptor. Let's say this time the acetylcholine binds to this receptor and causes it to open, right? So now we get an influx of calcium here, just a millisecond, for example, before another action potential arrives here and causes additional influx of calcium. So now we have more calcium than normal in this presynaptic neuron, right? Because these other channels that are permeable to calcium were opened. So now we have more calcium entering. That leads to more neurotransmitter being released from this presynaptic cell number 2. So now there's more neurotransmitter released than normal because there's more calcium in there, more binding than to these receptors on the postsynaptic cell, and that means we get more sodium entering through this postsynaptic cell. So this is called a presynaptic facilitation, because an arrival of an action potential at this presynaptic neuron resulted in more neurotransmitter release and more ion entry into this cell. Now, of course, this presynaptic inhibition and facilitation, really, it makes sense if you're working just with, uh, for example, channels that are permeable to calcium or sodium, right? But you could, but the same thing can happen for channels that are permeable to chloride or to potassium. If, for example, they are blocked, you would get more of an excitatory potential, and if they are if they're inhibited, you'd get more of a potential. If they're excited, you get less of a excitatory potential in this postsynaptic cell. So it really does depend on the receptor. But in this case, for facilitation, we get more neurotransmitter is released from the presynaptic neuron due to facilitation, or sort of an increase of signal transduction, right? And this is a little bit, it's different than how we've kind of thought about things before, because we've talked about action potentials being all or nothing responses, and it doesn't matter the size of the stimulus. Once an action potential fires, it's always the same strength and the same frequency, and it travels all the way down the cell. And that's totally true. And that's because we're not really talking about action potentials here. We're talking about graded potentials, right? And getting a cell to threshold. And of course, we remember that graded potentials are dependent on the strength of the stimulus. They do spread passively. They don't reinitiate themselves as they move across the membrane or across the soma of this cell. And they do diminish over time. So, and the strength diminishes over time. So that's why we can get inhibition or facilitation of the firing of an action potential by altering the intensity of the graded potential. Right? Okay, so please let me know if you have any questions about presynaptic inhibition or facilitation, and I'll see you in a future video.